So we say for this semester, we're talking about bacteria first. The first class, we're talking about general bacteria. The second class, we're talking about clinical bacterial infections. The clinical bacterial part. And the last class, we're talking about general virus. And today is the fourth class, we're talking about clinical virology. So that means we're talking about individual viruses, and then they cause the illness. So basically, we pick up some common illness and cause the illness. Certainly, we have much more viruses cause the different kind of illness, but we only have one day for special virus illness. So we pick up some common ones. I just updated our notes on my website this morning. I added COVID-19 in there. Yeah. The others is the same. Yeah, I just add COVID-19 because right now it is so important for look like everybody is severe kind of virus infection. So I just added this part. Yeah, if your notes don't have that part, you can reload it. Yeah, reload it. You will got this part. So COVID-19. So that's a virus illness. And they have another name, SARS-CoV-2. SARS-CoV-2. SARS-CoV, that happens in 2003. Still got like a lot of people infection in that year, but not like this year. Yeah. But still severe illness. The symptoms pretty similar, like a cough, fever, tired, severe case, pneumonia. Yeah, symptoms pretty similar. And the virus structure pretty similar also. Yeah. Just a little bit changing. Yeah, just a little bit changing there. So that's why it's normal or always name. Yeah, always name. But generally, right now, we just call it COVID-19 because it found in last year, 2019. Last year we found it. Yeah, last year we found it. And the COVID, just the shape of the virus. Yeah, it's the shape of the virus. So this virus belong to that group. We have group of virus. The shape pretty similar, kind of round shape. And you can see some smaller here. So this is the truly picture coming from an electrical stop. Yeah. Truly picture. Yeah. That's COVID-19 virus here. COVID-19 virus here. So this virus is a single line of the RNA virus. Yeah. Single line RNA virus. With a virus, could we have RNA, could we have DNA, but not have both. So that's the RNA virus. And it can spread between the animals and the humans. In this group, we say all the viruses have pretty similar shape. but will be a little bit different. There is a big group there. Right now, we find 7 of them. Yeah. They can infection for the humans, can cause the people illness. This one is the 7th. Yeah. Before this one finding, we only have 6, but right now we have 7. So we still go for radio to look at COVID-19.
Let's talking about COVID-19 virus today. COVID-19 virus, also called SARS-CoV-2 or new COVID virus. COVID-19 is pandemic in the world in 2020. Since December 31, 2019 and as of July 13, 2020, 12,875,963 cases of COVID-19 have been reported, including 568,628 deaths. Biology of the COVID-19 virus. COVID-19 virus belongs to coronavirus group. It is a kind of zoonotic single-stranded RNA virus that spreads between animals and humans. Coronavirus can infect mammals and birds, causing digestive tract diseases of cattle and pigs or upper respiratory tract diseases of chickens. 7 kinds of coronaviruses are common in nature and known to infect human beings. They can cause respiratory tract infections, common cold, and even severe diseases such as Middle East Respiratory Syndrome, MERS, severe acute respiratory syndrome, SARS, and new coronavirus diseases, COVID-19. In 2003, an epidemic caused by the SARS virus caused the term coronavirus to gradually enter people's vision. Coronavirus is named because it can observe the obvious accumulation of rod-shaped particles under the microscope. And the shape is like the crown of the medieval European emperor. In 1975, the Virus Nomenclature Committee officially named the coronaviridae. Coronavirus is an enveloped RNA virus that is widespread in humans, mammals, and birds, and can cause respiratory, infection, liver and nervous system diseases. There are 7 of coronaviruses could cause humans illness. 4 of them are currently known to infect humans, may cause common cold symptoms. 3 other viruses will cause severe illness, like severe acute respiratory syndrome coronavirus, SARS-CoV, Middle East Respiratory Syndrome virus, MERS-CoV, is a zoonotic virus that can cause fatal diseases, and severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, which also commonly be called COVID-19, since it be found in 2019. Coronavirus particles are irregular in shape, with a diameter of about 60 to 220 nanometers. The virus has an envelope structure with 3 proteins on it. Spike glycoprotein, S, spike protein, small envelope glycoprotein, E, envelope protein, and membrane glycoprotein, M, membrane protein. And a few types have hemagglutination vegetarian glycoprotein, He protein, hemagglutininasterase. Coronavirus is characterized by being able to use itself as a template to guide the synthesis of virus-related proteins. After the virus enters the host cell, RNA polymerase is first expressed using viral RNA as a template, and then RNA polymerase completes the transcription and synthesis of negative strand RNA, the synthesis of various structural protein RNAs, and the replication of viral genomic RNA. 2019 nCoV. In December 2019, China discovered a group of patients with pneumonia caused by unknown reasons related to the seafood wholesale market. A new coronavirus was detected by whole genome sequencing. The virus belongs to the subgenus SARS-virus, or Orthocoronavirus subfamily. This virus is different from MERS-CoV and SARS-CoV. It becomes the seventh member of the coronavirus family that can infect humans. Virus morphology. This transmission electron microscope image shows SARS-CoV-2, also known as 2019 nCoV, the virus that causes COVID-19. Isolated from a patient in the U.S. Emerging from the surface of cells cultured in the lab. The viruses were generally spherical. The diameter is between 60 to 140 nanometers. Virus particles have obvious spinous processes, about 9 to 12 nanometers, causing the virus to exhibit a coronal shape. 2019 nCoV infection of host cells. For a virus to enter a cell, the cell must have its corresponding receptor. For example, the common receptor for HIV is CD4 protein, which is usually on the surface of immune cells in the blood. So HIV can be transmitted through the blood without worrying about airborne transmission. The receptor of this new coronavirus is angiotensin-converting enzyme 2 like SARS, which means that to infect humans, the virus must first contact cells with this enzyme to complete receptor binding. And we happen to have many of these cells exposed to the air, the mucosa. The meaning of the mucosa is to secrete mucus and keep it moist. There are a lot of mucosal cells in our lips, eyelids, nasal cavity, and mouth. When the virus contacts the oral mucosa in some way and binds to the receptor, the infection begins. First, the envelope of the new coronavirus will fuse with the cell membrane to release the viral genetic material, a single strand of RNA, which can be used as messenger RNA to deceive the ribosomes in the cell. Synthesize RNA replicase. RNA replicase will be based on viral RNA generates negative RNA strands. This negative RNA strand will continue to generate more RNA fragments and positive RNA strands with replicase. These different RNA fragments will generate more different viral protein structures with ribosomes. Finally, these proteins, the outer shell, will combine with RNA to generate new coronavirus particles, which are secreted out of the cell through the Golgi apparatus and infect new cells. Each infected cell produces tens of thousands of new virus particles that spread to organs and bronchial tubes and eventually reach the alveoli, causing pneumonia. New coronavirus, simple structure, simple composition, but destructive power is never simple. COVID-19 is usually excreted through tiny particles formed by the patient's cough, sneeze, or breathing. The droplets usually fall to the ground or onto surfaces rather than traveling through air over long distances. Transmission may also occur through smaller droplets, called aerosol, that are able to stay suspended in the air for longer periods of time. Aerosol transmission can happen during some medical procedures and potentially in crowded indoor spaces that are inadequately ventilated. Less commonly, people may become infected by touching a contaminated surface and then touching their face. It is most contagious during the first 3 days after the onset of symptoms, although spread is possible before symptoms appear, and from people who do not show symptoms. Common symptoms include fever, cough, fatigue, shortness of breath, and loss of sense of smell. Complications may include pneumonia and acute respiratory distress syndrome. The time from exposure to onset of symptoms is typically around 5 days but may range from 2 to 14 days. Diagnosis. The standard test for current infection with SARS-CoV-2 uses RNA testing of respiratory secretions collected using a nasopharyngeal swab, though it is possible to test other samples. This test uses real-time RRT-PCR, which detects the presence of viral RNA fragments. Imaging. Chest X-ray examination. Since the disease manifests itself as pneumonia, radiological imaging has a fundamental role in the diagnostic process. Standard radiographic examination, X-ray, of the chest has a low sensitivity in identifying early lung changes and in the initial stages of the disease. At this stage, it can be completely negative. In the more advanced stages of infection, the chest X-ray examination generally shows bilateral multifocal alveolar opacities, which tend to confluence up to the complete opacity of the lung. Pleural effusion can be associated. Chest computed tomography. Given the high sensitivity of the method, chest computed tomography, CT, in particular high-resolution CT, HRCT, is the method of choice in the study of COVID-19 pneumonia, even in the initial stages. There is no known vaccine or specific antiviral treatment. Primary treatment is symptomatic and supportive therapy. Recommended preventive measures include hand washing, wearing a face mask in public settings, maintaining distance from other people, covering one's mouth when coughing, disinfecting the surfaces, increasing ventilation and air filtration indoors, and monitoring and self-isolation for people who suspect they are infected. I hope everyone can take good protection and let this epidemic end soon. Thanks.
So that's the general information we're talking about COVID-19. That's coming back. So COVID-19, the office name, SARS-CoV-2, because the pretty similar structures with SARS-CoV virus, but little bit different. It's little bit different. And that's the picture. The picture is not truly photo, but is basically show us the covers kind of virus, what they look like. They are hardly to separate, like how clear, like SARS virus and COVID-2 virus. But that's generally look like this. Round shape, like a ball, and pretty small diameter, 60 to 140 nanometer, but still tiny smaller there. And they have some spawner. It's like this part. Like this part, spawner. And that's basically look like a corner shape. So that's the name coming from. Transmission. We say it transmissions by droplet. Pretty tiny small. Droplet. So usually, coming from patient's cough, sneezing, or even from the breathing. It's depending, like how many virus in the patient's mouth and nose, and even bronchitis, or even in the lungs. So they say the illness, infections, transmission, basically we will have three parts. One part is the passenger coming from. Usually is the patient. The second part is transmission ways. How the illness can transmit. And the third part is the patient or human being or people, or maybe we just use hospital, can receive it. So that's the three part mainly transmission kind of illness. So certainly, if we don't have pathogen, we don't have special kind of bacteria, we don't have special kind of illness, nothing can transmit. Secondly, it must be have some special way they can transmit from one people to another people. If we stop the way they transmit, they will not transmit for another people. And the people or host need more protected. So we say COVID-19 is transmitted by droplet, by people's cough, by sneezing or breathing. So that's why right now we try to protect each other. We're talking about. That's why we're talking about the mask, face cover, face cover. Maybe both for patients and maybe for other people. That's the way we try to protect it for ourselves. So we say these kind of small things coming from the patient's cough, sneezing, or breathing, and from the droplet. So usually they will fall down to some surface. It's not traveling that long distance. So that's why they keep the social distance. But truly, some pretty small droplets that maybe can stand in the air and maybe travel a little bit longer of time. So that is the more dangerous part. So also some transmit away. We say mainly transmit away by the air, by the droplet. Another transmit away may happen. We're talking about during some medical process. So that means like the hospital, if they are not protected properly, maybe still can transmit this kind of virus from one patient to another people. And also, more dangerous situation if a lot of people together, especially indoor space. Certainly, outdoor space, if a lot of people together, still can go to transmit. So that's basically transmission way. But occasionally, some people touching, we say some droplet go to some superficial space there, and some people touching some superficial stuff, like the desk, or even like on the bus, or even you go to shopping or whatever, you touching some space, superficial there, is being contaminated by the patient. It must have virus in there. And then touching the face. So face is pretty close to our nose and the eyes also. Then easily got the infection. So that's the transmission rate. But mainly still through the air. The common symptoms. So people got like a transmission by others, got a virus infections for COVID-19. So we're still talking about COVID-19 here. Certainly different kind of virus infection, they may be different. But for COVID-19, totally some people maybe know any of the symptoms. Even themselves maybe don't know they got COVID-19 infection. So that's especially. So that's still we're talking about before. It's depending the immunity functions and how severe the passenger kind of benefit. The patient got symptoms usually after they touching the virus or got virus infections, usually after 2 to 3 days. And then some symptoms may show up. So the typical symptoms, patient may fever. Yeah, patient may fever. So we say fever itself is our body protected for body weight, but truly causes people uncomfortable. And patient will be fatigued, tired. And the typical symptoms, patient will be cough because the virus will invading. They go in from our nose or maybe mouth, and then go to our throat area, then go down to the trachea, then go to throat and the trachea area, and cause our body reaction. So some fluid will come in, and with the fluid, some like specially or immunity reaction in there, like we talked about, some special kind of cells in there, and special kind of protein will be there, and try to against the virus. So in that time, will cause the patient cough, especially in the trachea. With the trachea will be an empty space. There will be an empty space. Some soft cartilage holding a space there, and some muscles in there. But it's truly pretty empty. It's only allow the air passing through. Anything in there will cause the people cough. So right now, some virus go to there, and some fluid or phlegm in there. So certainly cause the patient cough, phlegm. So when people cough, some phlegm coming out, some virus coming out. So that's basically the transmission way through the patient's cough. And when the virus go to the lung, when the virus go to the lung, basically cause pneumonia. So pneumonia is the virus go to the lung, and the pneumonia basically virus in there and some fluid in there. Certainly air couldn't go in. If the air couldn't go in, people will be short of breath. So these two symptoms pretty closely related. And then that's a severe kind of situation will be happen. ARDS. That's acute respiratory distress syndrome. Means the patient's severe kind of short breath, and the air changing will be pretty decreased, and the oxygen level in the blood circulation is decreased. So that's pretty severe kind of conditions. People may be half died from this. Yeah. So that's we heard about some people died from COVID-19. That's basically they cause the acute respiration severe situation first, and then no oxygen. Yeah, no oxygen. So no oxygen supposed to the organs, all the organs, you know, and then people will die. Yeah. And some special case, some special case people possible losing the smelling function. Means some virus especially damage the nervous system in the nose. And also we heard about some people, they may losing the tasting function. So that means damage the nervous system in the tongue, or tasting relationship of tongue. And we have some tasty body here. So special bacterial invading that part. So that's some common symptoms for this one. So diagnosis. So diagnosis right now, like the radio, we can see we use a pretty long strip and take some sample from back part of the nose, landing area. So that's collected some sample, and then with that sample we do some tests, some special RNA tests, because that belongs to RNA virus. So then we can find. But truly, it's not 100% if people got infections, 100% positive. So it's totally depending. How many viruses in there, and also depending the patient take the sample. So we say in the third area, basically the pretty deep area of the nose area. We take a swipe, right, go to there and take a sample there. So sometimes maybe not that effective, sometimes maybe not deep enough, or maybe the patient mainly cause the infection of the lung, not too many viruses in there. So only some of them they got positive reactions. Some of the patients even not reacted with it, like we repeated the test sometimes after 10 days, they may be changing from negative to positive. That means more virus in production in that area. So that's the basic way right now we use for diagnosis. But if some people got negative reactions, they couldn't 100% to say he didn't got infection. But if positive, that looks like 100% diagnosis in there. They have some other ways to check to make diagnosis. One way is X-ray check. X-ray check usually pretty simple, and easy to perform. Let's take X-ray pictures like the video show us. So basically, if you're looking for X-ray pictures, if the dark color is normal, dark color is normal. Dark color means it's air in there, So we can see some white color, that's the rib, But between the rib and the rib, that's dark color or black color. So that's the normal color for X-ray for the lung, If you see some part, it's more white color. That means in that area, more fluid in there, or sometimes tumor in there. But for COVID-19, it's fluid in there, it's pneumonia. So that's we try to find through the X-ray. Like we say, X-ray is easy to check in, but for early stage, it's not that sensitive. Maybe view normal. Another more porous way, CT scan. they have different level of CT right now. Certainly high level is more effective, more sensitive. So CT scan basically they still belong to emerging, like some picture in there, but they cut the tissues, about 1 centimeter or even more smaller with high effective CT. So that means something about 1 centimeter they still can see. But for X-ray, usually they must be much bigger, sometimes 3 to 5 centimeters, maybe we can see it. Otherwise, look like normal. So that's another way to make diagnosis. We combination together right now. So like people got the raw simple, not find anything, but patient have symptoms, go to CT checking or maybe X-ray checking. Immunity part, we talking about with the immunity, we including active and positive. We don't have vaccine right now available now. That means active part we don't have yet. It takes time. I know a lot of researchers. They try to like make some vaccine available for the people, but right now it's not got the result yet. Usually they take some time. It's not that easier. Usually a few months or even a few years, but they are working for that. They are working for that. But right now still not have yet. Still not available. So what's the illness totally could be controlled? Vaccine. We have a lot of illness like 100 years ago. In that time, it's very severe. But later on, when they have vaccine, it's controlled. Right now, COVID-19 is not truly be controlled because we don't have vaccine. We don't have active kind of immunity. Positive immunity, we say use antibody. So antibody part usually coming from like animals or maybe coming from producing by some factories. Right now, that part we don't have yet. We still don't have that part yet. But truly clinically, like some people getting sick or COVID-19 infections, and then they recover, totally recover. So these kind of people, in their blood, they will have antibody. They will have antibody. So these kind of patients, they recover. They have antibody. They can donate their blood for the patient. They have severe kind of infections and can save the people's life. And it truly happens right now. Some people got infections and then they recover and then they donate their blood to treat some severe kind of patients. That part, right now we use. So that's the immunity part. Treatment part. So treatment part, we don't have specially kind of anti-COVID-19 virus medication right now. Not effective kind of antivirus medication. Some antivirus medication, like anti-other virus, kind of benefit for some of the patients, but not for all the patients. So mainly treatment right now, relieve the symptoms, and also build up the patient's immunity function, like the patient's immunity, they performing function and against the virus, and then cure it. So that's why a lot of people, they got sick. They didn't go to special, like go to hospital to treat. We talk about it, stay at home, maybe drink a little bit more water, and eating a lot of nutrition, and rest, and then they recover. Most patients recover by itself. But truly, if patient have severe kind of situation, like pneumonia or even ARDS kind of condition, must be go to hospital. Because their oxygen level is pretty low. Need like give the patient more oxygen and support the organs function also. But it's not that severe. Like there's a cough, fatigue, and lose some memory function or tasting function, even not severe kind of fever. Maybe patient they stay at home. Because right now we don't have special kind of treatment for these patients. Preventing is more important. Preventing is more important. So like now we don't have vaccine. We don't have effective kind of treatment. So more important, preventing. Don't get it. So we know that mainly transmission way by the air. So put a face mask, especially when you go to a lot of people in there, and especially inside of the door, even outside of the door. If a lot of people in there, still better put a face mask or face towel. And certainly keep the distance from other people. So right now we're talking about the 2 meters kind of social distance away from other people. And hand washing. Some of the transmission way we're talking about we cutting the superficial, like the desk or some material in there, and then put your hand in your face, maybe cause the transmission. And always cover the mouth, the cough, sneezing. That's protected for other people generally. So right now, we say some patients, they don't know they have virus infection, no symptoms. So everybody try to protect each other. And disinfecting the superficial, to kill the virus, and changing the air. So air ventilation. The good way, open the door, open the window. Air condition right now is not working that well because air condition, especially for central control kind of air condition, they circulate the air from one room to another room. So in that way, changing the air, it looks like some risk. They travel the virus from one room to another room. So we say open the window, open the door. That's the best way. And certainly, if some people getting sick or maybe suspected they got infection, we got isolation. So that's the first virus we're talking about right now. It's more concerned for most people for COVID-19. So any question about that? Then we go to next one. Yeah. Hyperspheres. Yeah. Hyperspheres is DNA virus. Yeah, it's DNA virus. It's development. Yeah, it's development. The DNA virus. Still a group. Yeah, still group. They have like type 1, type 2, another special one we call VZV. So this one clinically cause no problem. So we mainly talking about this one. So this one, that's the one virus, yeah, the same virus, but they cause the two different illness. One illness name we call chickenpox. Chickenpox. That's the mainly people first time infected by this virus. Another illness we call single. That's the same virus. Secondly, cause the illness. So that's the same virus, but they cause the different illness. Let's still go to our video to see it.
Let's talking about herpes viruses today. Herpes viridae is a large family of DNA viruses that cause infections and certain diseases in animals, including humans. The members of this family are also known as herpes viruses. The family name is derived from the Greek word herpine, referring to spreading cutaneous lesions, usually involving blisters, seen in flares of herpes simplex 1, herpes simplex 2, and herpes zoster, shingles. All members of the herpes viridae share a common structure, a relatively large, double-stranded, linear DNA genome encoding 100 to 200 genes encased within an icosahedral protein cage called the capsid, which is itself wrapped in a protein layer called the tegument containing both viral proteins and a lipid bilayer membrane called the envelope. Varicella zoster virus. Varicella, chickenpox, is the primary disease. Zoster, shingles, is the recurrent form. Chickenpox, also known as varicella, is a highly contagious disease caused by the initial infection with varicella zoster virus, VZV. The disease results in a characteristic skin rash that forms small, itchy blisters, which eventually scab over. It usually starts on the chest, back, and face. It then spreads to the rest of the body. Other symptoms may include fever, tiredness, and headaches. Symptoms usually last 5 to 7 days. Complications may occasionally include pneumonia, encephalitis, and bacterial skin infections. The disease is often more severe in adults than in children. Symptoms begin 10 to 21 days after exposure to the virus. Chickenpox is an airborne disease which spreads easily from one person to the next through the coughs and sneezes of an infected person. It may be spread from 1 to 2 days before the rash appears until all lesions have crusted over. It may also spread through contact with the blisters. Those with shingles may spread chickenpox to those who are not immune through contact with the blisters. The disease can usually be diagnosed based on the presenting symptom, However, in unusual cases it may be confirmed by polymerase chain reaction, PCR, testing of the blister fluid or scabs. Testing for antibodies may be done to determine if a person is immune. People usually only get chickenpox once. Although reinfections by the virus occur, these reinfections usually do not cause any symptoms. Signs and symptoms. A single blister, typical during the early stages of the rash. The early symptoms in adolescents and adults are nausea, loss of appetite, aching muscles, and headache. This is followed by the characteristic rash or oral sores, malaise, and a low-grade fever that signal the presence of the disease. In children the illness is not usually preceded by prodromal symptoms, and the first sign is the rash or the spots in the oral cavity. The rash begins as small red dots on the face, scalp, upper arms, and legs, progressing over 10 to 12 hours to small bumps, blisters, and pustules, followed by umbilication and the formation of scabs. At the blister stage, intense itching is usually present. Blisters may also occur on the palms, soleus, and genital area. Commonly, visible evidence of the disease develops in the oral cavity and tonsil areas in the form of small ulcers which can be painful or itchy or both. These symptoms of chickenpox appear 10 to 21 days after exposure to a contagious person. Adults may have a more widespread rash and longer fever, and they are more likely to experience complications, such as varicella pneumonia. Because watery nasal discharge containing live virus usually precedes both exanthem, external rash, and enanthem, oral ulcers, by 1 to 2 days, the infected person actually becomes contagious 1 to 2 days before recognition of the disease. Contagiousness persists until all vesicular lesions have become dry crusts, scabs, which usually entails 4 or 5 days, by which time nasal shedding of live virus ceases. The condition usually resolves by itself within a week or 2. The rash may, however, last for up to 1 month. Chickenpox is rarely fatal, although it is generally more severe in adult men than in women or children. Non-immune pregnant women and those with a suppressed immune system are at highest risk of serious complications. Diagnosis: chickenpox. The diagnosis of chickenpox is primarily based on the signs and symptoms, with typical early symptoms followed by a characteristic rash. Confirmation of the diagnosis is by examination of the fluid within the vesicles of the rash, or by testing blood for evidence of an acute immunologic response. Vesicular fluid can be examined with a Zank smear, or by testing for direct fluorescent antibody. The fluid can also be cultured, whereby attempts are made to grow the virus from a fluid sample. Blood tests can be used to identify a response to acute infection, IgM, or previous infection and subsequent immunity, IgG. Prevention. Hygiene measures. The spread of chickenpox can be prevented by isolating affected individuals. Contagion is by exposure to respiratory droplets, or direct contact with lesions, within a period lasting from 3 days before the onset of the rash, to 4 days after the onset of the rash. Vaccine. The varicella vaccine is recommended in many countries. Some countries require the varicella vaccination or an exemption before entering elementary school. A second dose is recommended 5 years after the initial immunization. A vaccinated person is likely to have a milder case of chickenpox if they become infected. Immunization within 3 days following household contact reduces infection rates and severity in children. Treatment. Treatment mainly consists of easing the symptoms. As a protective measure, people are usually required to stay at home while they are infectious to avoid spreading the disease to others. Cutting the nails short or wearing gloves may prevent scratching and minimize the risk of secondary infections. Shingles. After a chickenpox infection, the virus remains dormant in the body's nerve tissues. The immune system keeps the virus at bay, but later in life, usually in an adult, it can be reactivated and cause a different form of the viral infection called shingles, also known as herpes zoster. Shingles affects 1 in 5 adults infected with chickenpox as children, especially those who are immune suppressed, particularly from cancer, HIV, or other conditions. Stress can bring on shingles as well, although scientists are still researching the connection. Shingles is most commonly found in adults over the age of 60 who were diagnosed with chickenpox when they were under the age of 1. The signs and symptoms of shingles usually affect only a small section of one side of your body. These signs and symptoms may include pain, burning, numbness, or tingling, sensitivity to touch, a red rash that begins a few days after the pain, fluid-filled blisters that break open and crust over, itching. Some people also experience fever, headache, sensitivity to light, fatigue. Pain is usually the first symptom of shingles. For some, it can be intense. Depending on the location of the pain, it can sometimes be mistaken for a symptom of problems affecting the heart, lungs, or kidneys. Some people experience shingles pain without ever developing the rash. Most commonly, the shingles rash develops as a stripe of blisters that wraps around either the left or right side of your torso. Sometimes the shingles rash occurs around one eye or on one side of the neck or face. Treatment. Treatment mainly consists of easing the symptoms. Antivirals are sometimes used. Acupuncture could help to relieve pain. Thanks for watching. If you
So that's the general information about this virus. So we still continue here. So we say these virus cause two illness. One illness is chickenpox. That's the first time people got this virus infection. And then most people will be cured. But for some of the people, the virus will be holding in patients' nervous system. And then when people's immunity deficiency time, they will show up still skin rash, but that kind of skin rash is follow the nervous passing route. So that's why we only see in half part of the body. Maybe left side, maybe right side, and most cases follow the rib. They follow the rib because the nervous travel under the rib. That's the second time, the second form. It's not like people got the second time the same virus infection. It's just the virus staying there and waiting the time. The transmission way. So transmission way is still by the air, still do that. And also sometimes if directly touching, we say these patients usually have skin rash in there. If we directly touching the skin rash area, virus will be there. So chickenpox is very popular illness for the children. It's high contaminant. But right now we have effective vaccine and protected. So look like right now we don't have many people or many children get chickenpox. But truly, when people got these virus infections, mainly skin area, we will have kind of skin rashing and even some small blister in there and some liquid inside. So that's generally say. So these varies, mainly cause the skin rash. Mainly cause the skin rash. And they also invading our nervous cells. Also invading the nervous cells. So we say they can hold in there for long term. When people's immunity function decrease, they cause the second infection. And also infection, the respiration tract. That's why we say that's still mainly transmitted by the air, similar like COVID-19. So that's mainly. When they got the secondly cause the illness, we say the virus holding there for long term. But when people's immunity function is decreased, or some other reasons cause the patients, like a local trauma or local weakness in there, and the virus can active again and still cause the skin rashing and viral symptoms. We say basically the invading nervous cells, so cause viral symptoms. Clinical symptoms. For chickenpox, basically, patient may have fever first, uncomfortable feeling, people feel sick. Usually after 2 or 3 days, skin rash will be show up. And the rash area will have some booster in there, more liquid in there, and even pops. And then gradually recover. So each during the healing process, some itchy. That's why people always try to scratch in there because itchy. So chickenpox is more itchy compared with pain. And usually this illness is not that severe. But if patient never got infection when they're young, but they getting older and then they got infection, that's truly is not severe. And severe case, we say most cases not severe, but truly possible got some severe case, especially for adults, maybe cause pneumonia, maybe cause the infection in the brain. So that's more complicated. Mostly happens in the audience. So there's some picture here. So like a chickenpox, they can go all over the body, in the trach area or maybe on the face and the chest area. But you can see that's on the all over the body. But the singles, we say that follow the nerve branch. They go and only go to one side of the body. And skin rash is different. Chickenpox look like mostly the single one in there, but the singles, like a lot of connected each other. So that's the difference. So treatment. So basically, it's still not necessary for special kind of treatment. If patient got the chickenpox, no effective. Severe kind of case, maybe we use some antivirus medication, but mostly recover by itself. That's basically certainly into severe case, they cause other issues. We still treat other issues, problems. And the immunity, we say it can got naturally immunity. If patient got chickenpox, they will not have got the chickenpox again. Only getting one. Everyone only getting one. Whatever you got the vaccine, or maybe you got the sick transmission from others, you only got one. But it's possible getting singles later. And the singles, most time only happens once, but still possible have multiple times. It's depending how like the immunity systems, how it's strong or weaker. Prevention. We say vaccine is available right now, and a lot of many countries have vaccine available. So that's the mainly the prevention way. And we say without vaccine in the children. So basically under 12 years old. And then second time, usually we do twice. Right. For the single vaccine, right now we think about some people immunity function is low, maybe got a single vaccine, especially people age over 60, and the immune system is weaker. And still be careful. The vaccine for the singles, we use the leaf virus. It's less toxic, but still leaf virus. For people with severe kind of immunity deficiency and Parkinson's women, their immunity generally is weaker. They need to be careful to use or maybe not use. So that's the virus relationship with chickenpox and shingles. Now we move into that one, hepatitis virus. So generally hepatitis virus, we talked about last semester already. We go a little bit faster. So hepatitis virus, totally we have 5 different kinds, but pretty commonly hepatitis A and hepatitis B. So that's 2 commonly caused illness. So hepatitis A caused by hepatitis A virus. When we talking about hepatitis, it means liver infection. Virus infecting the liver. Transmission way from the stool to the oral routine. So we say no people eating the stool, but just touching, sometimes just touching, like not washing the hand properly. And after touching, usually after 2 weeks, then people will have some symptoms. So that's usually we say that's the transmission way. Certainly, easily cause the infection in the summer, like the people in the summer. it's not that pay attention. And also it causes illness, like a lot of people eating in the school for the children. And transmission may usually coming from the water or the food. Hepatitis A virus usually not transmission by the blood, but occasionally. So that's hepatitis A. And clinically symptoms pretty similar like other hepatitis. We're talking about patient maybe fever, maybe poor appetite, maybe nausea, vomiting, and jaundice, yellow skin, yellow eyes. And dark urination, pale stool. And if we go to blood test, so we can find special kind of adenine will be, the number will be increased. So hepatitis A can recover by itself. So usually 2 to 4 weeks. And we say people got hepatitis A infections, and they may not immediately go to illness. They may after 2 weeks or even 3 to 4 weeks, then they got illness. Hepatitis A is acute one. Yeah, it's acute one. So usually it's cured. It's not changing to chronic. So basically, if they are not changing to chronic, they will not cause the other problems. And diagnosis, usually people have symptoms and then go to laboratory diagnosis. We have a special kind of immunity protein we call IgM. We can find special kind of antibody that can make a patient diagnosis. Treatment, we say various kind of illness. Usually we don't have effective kind of medication or drugs to treat. So basically, it can cure it by itself. So that we say for most cases, people just get some rest, get good nutrition, and then they recover by itself. And immunity. We have effective vaccine. So hepatitis A, we can vaccine. So basically, we vaccine in the children time. Yes, still in the children time. And if we talk about it takes time to active kind of immunity takes time to like getting it working. But if some people, especially like they didn't got the vaccine before, but they did travel to some especially academic kind of area, in this kind of situation, we give the patient positive kind of immunity, means give the patient antibody directly. So combination kind of positive and active kind of immunity. So that's hepatitis A.
Sorry, could you just go back for 1 second? Just one slide.
Okay, yeah.
Sorry. Okay, I got it. Thank you.
Yeah. Then we talking about hepatitis B. Hepatitis B caused by hepatitis B virus. The name look like similar, but they truly different virus. yeah, different virus, because they both cause the liver infection. So that's why they name it. Transmission way for hepatitis B virus mainly is blood. Mainly is blood. Like people use the same needles, it's truly transmission hepatitis B. And also maybe by some sex activity. And also from mom to the new baby, like the process, the baby delivery. So the symptoms, symptoms. Some people got hepatitis B infections, they may be totally no symptoms. Virus, they have virus. They can transmit to other people, but they don't have symptoms. Or maybe they recover. Yeah, they have hepatitis B, they don't know, they recover. Later on, we can find the antibody. If we find some special kind of antibody for some people, that means they must have got this illness before. But they don't know. They didn't have any symptoms. So clinical symptoms pretty similar like hepatitis A. We still talking about fever, nausea, vomiting, low appetite, jaundice. Yeah, symptoms pretty similar. But maybe that be severe. And hepatitis B truly can change into chronic process. They may like recover from acute state, look like no symptoms, but their virus still in the body. And some people maybe have chronic symptoms. So these kind of patients maybe cause the cirrhosis later. We talk about cirrhosis later also. And for fabricated changes in the liver. And maybe lending patient death. So for diagnosis, we're still talking about we got like the symptoms, the symptoms. Then we got laboratory tests for some antigen, hepatitis B antigen, and some antibody. So that's for laboratory tests and for the treatment. So still not that effective treatment for hepatitis B. We have some medication. But not that effective. We can use, but it's not working that well. Preventing. So still mainly we preventing vaccine. Or maybe we use both active kind of immunity or positive kind of immunity same time. If we're talking about hyperimmunity globin is antibody. So that's basically for hepatitis B. But truly hepatitis B look like cause the most severe problems compared with hepatitis A because that's a chronic process and a lot of people don't have symptoms, but they also carry virus. So especially for we do needles, pay attention some people if they have hepatitis B infections. So pay attention, don't use needles, transmission, that kind of virus to other people, or maybe protect it for ourselves for needles access. So that's hepatitis B. HIV virus.